Bismillahirrahmanirrahim. Assalamu alaikum. My name is Ibrahim and I am a student of the Field Microbiology at Cohort University of Science and Technology, Cohort. This is my presentation topic for abstract presentation is control of root knot nematode, Merodegan incognita, in common bean plant by Trichoderma species isolated from the fields of district Cohort. Content of my presentations are introduction, rationale, objectives, material and methods, results, conclusion and recommendation, and references. Introduction. Common bean, Phaseolus vulgaris, is a member of the family Fabaceae and contains low fats, high protein contents, dietary fibers, and complex carbohydrates. In Pakistan, due to various reasons, including nematodes infestation, the production of common bean is insufficient to meet the requirements. For the control of plant pathogenic nematode, Trichoderma species considered as a suitable biocontrol agent. Biocontrol reduce the reliance on synthetic pesticides, reduce the adverse effect on the environment, improves worker safety, and maintain the economic viability of crops. Rationale. Therefore, the present study was based on isolation of nematocidal strain of Trichoderma species from the fields of district Cohort against the root knot nematodes carrying disease in common bean plant. Objectives. Objectives are to isolate and identify the Trichoderma species from the fields of district Cohort, to extract and characterize root knot nematode from the root of common bean plant, and to investigate the nematocidal activity of Trichoderma species using the in vitro and in vivo bioassay. Material and methods. In material and method, first of all, soil samples were collected from different locations in district Cohort. So were proceed. Then next, a serial dilution were prepared. For the isolation of Trichoderma species, the serial dilution were poured on two different media, potato dextrose agar and Trichoderma selective medium, and incubated at 25 degrees centigrade temperature for 3 days. After incubation period, the suspected Trichoderma colony were subcultured on PDA and were subjected for morphological identifications. The morphological identification were based on colony morphology and microscopic characteristics. Isolation of Merodegan incognita. Root samples were collected from infected common bean plants. Then nematodes were isolated by the pollen method from the and root incubation method from the common bean roots. Then the isolated nematodes were identified morphologically and morphometrically. The identified nematode were then propagated on common bean plant grown in greenhouse conditions. In vitro and in vivo nematocidal activity of Trichoderma. During in vitro nematocidal activity of Trichoderma conidia suspension and culture filtrate of Trichoderma were prepared. Then the nematodes suspension were prepared by disinfection it and prepare for the in vitro activities. Then 1 ml of conidia suspension and culture filtrate transferred to a petri plates containing 50 juveniles or eggs per 10 microliters. During in vivo nematocidal activity of Trichoderma against root knot nematode Merodegan incognita, Trichoderma conidia suspension were prepared. Then the seed of common bean plant were sterilized and treated with Trichoderma conidia suspension. The treated seeds of common bean were planted in a pots containing 1,000 nematodes per pots. After 8 weeks, the different plant characteristics were measured and then statistically were analyzed. Results. Total 5 soil samples were investigated for isolation of Trichoderma species. In all the samples were positive for Trichodermas. The figure 1 shows microscopic pictures and colony pictures of the isolates. The upper rows are the microscopic picture, while the middle and below rows are the front and back side picture of Trichoderma isolate colonies. Out of 5 isolate 3 were selected based on in vitro activity for in vivo and morphological and molecular identifications. Table 1 shows colony characteristics of trichoderma isolates on PDA and SNA, synthetic mutant agar. All three isolates have shown different characteristics on different media on PDA and SNAs. Characteristics such as color of the culture, colony, form of PDA, conidiation, and pigmentation were noticed. Microscopic characteristics of trichoderma isolates. These are the microscopic characteristics of trichoderma isolates. The microscopic characteristics such as shape and size of conidia, phyllides, presence or absence of striate hair, and presence or absence of dermatospores were noticed. All the isolates were noticed to produce all the isolates were noted that the striate hair were absent in all the isolates, while dermatospores is present only in the isolate 2. Morphological and morphometrical data of merodecarn. This table shows the different characteristics of merodecarn and cognata. And characteristics includes body shape, body length, neck length, stat length, knob shape, stat knob length. Table 3 shows different characteristics of merodecarn and cognata. These characteristics includes body shape, body length, neck length, stat length, knob shape, stat knob length, interior into exterior fold, tail lengths of female and male merodecarn and cognata. These characteristics were previously also reported by Carson et al. Figure 2 shows the trichoderma conidia suspension and cultural rates on J2's motility. The highest activity of trichoderma isolates were noted against J2 motility in the isolate 3. Conidia suspension shows 68% of the J2's motility, while in culture filtrates J2 motility were 71%, and so on. Figure 3 shows the effect of trichoderma conidia suspension and culture filtrate on egg hatchings. The egg hatching inhibition, the highest egg hatching inhibition was noticed in isolate 3. Culture filtrate shows 68% of the egg hatching inhibition, while the spore suspension has shows 58% of the eggs hatching inhibitions, and so on in isolate 1, isolate 3, isolate 4, isolate 5, and isolate 3 were the most effective. Figure 4 shows the effect of trichoderma on galling and egg mass index in the common bean plants. These were the in vivo studies during which the highest activity of trichoderma isolate 3 were noticed. Trichoderma isolate 3 has significantly reduced the gall index and egg mass index in common bean plant and has also shows the plant growth promoting traits. Figure 5 shows the antagonistic activity of trichoderma against plant pathogenic fungi. The highest activity of trichoderma isolates were noticed in isolate 3. The fungal pathogen were Aspergillus niger, Rhizoctonia solini, and Fusarium aureus. The highest activity against Rhizoctonia solini were noticed about 25%, while against Fusarium aureus, the activity of trichoderma isolate 3 were 73%, and against Aspergillus, the activity were 58%. Conclusion and recommendation. From the study, it is concluded that trichoderma isolate ISO3 has shows a significant toxicity against merodecarn cognata during in vitro and in vivo both studies. Further, a large-scale study is required to find the efficacy of above isolate in the fields. These are the references. Thank you for the attentions.